So the final talk of the evening will be Gene Winter from the National Human Genome Research Institute in Bethesda. And Gene will tell us about a systems genetics approach to prostate cancer.
Thanks very much. I'd like to thank the organisers for allowing me to talk today, and thanks everyone for sticking around. If Steve didn't put you to sleep, hopefully I will. So I'm a first-year visiting postdoc fellow at the NHGRI, and I'm in the Crawford lab, and we're interested in looking at hereditary variants and their influence on susceptibility to aggressive prostate cancer. And I'll be talking to you today about analysis of germline modifiers of aggressive prostate cancer using a cross-species variation and systems genetics approach. So why prostate cancer? Well, it's a common malignancy in men. In 2012, over 1.1 million cases were diagnosed worldwide, and there's a lot of cases in more developed regions. In the US, there'll be over 180,000 new cases diagnosed in 2016. So the risk factors are both environmental and genetic. However, only about 14% of all cases result in death directly due to prostate cancer. So there's a lot of men that have prostate cancer but are dying from other causes. So there's a lot of overtreatment of this disease. And at diagnosis, a very small percentage, less than 1% of prostate cancer cases are a subtype of prostate cancer called neuroendocrine prostate cancer. But at the time of death, the amount of cases mortality cases jumps to 25%. So we're really interested in identifying the development of neuroendocrine prostate cancer. So overwhelmingly, about 90 to 95% of prostate cancer cases are adenocarcinoma. They develop through a series of somatic changes in the cells which leads to prostatic intraepithelial neoplasia and eventually adenocarcinoma. And this is androgen dependent. And although it can metastasize, it's not usually fatal. So because it's androgen dependent, the first line of treatment is usually androgen deprivation therapy. And while this works for a few years in most men, after a couple of years, a majority of men actually regress and develop castrate-resistant prostate cancer. And this can lead to neuroendocrine carcinoma. We also can see adenocarcinomas with histological characteristics of neuroendocrine carcinoma. And what's important about this is that it's very aggressive and metastasizes a lot, particularly to visceral organs such as the liver and lungs. Neuroendocrine carcinomas genetic characteristics by loss of RB and increase in MYC-N. So we're really interested in defining the molecular characterization of the development of neuroendocrine carcinoma. So one mouse model that we use that's really useful in studying neuroendocrine carcinoma is the TRAMP mouse model of prostate cancer. It has the SV40T antigen, which is a oncogenic protein, and it's driven by the androgen-regulated probasin promoter. And this is prostate-specific. So after the onset of puberty, we see development of prostatic hyperplasia and neoplasia. And by 30 weeks in the C57 black 6 background, we see 100% of mice develop prostate tumours by 30 weeks. And at 28 weeks, we see metastases to the liver, lungs, and lymph nodes. But what's great about this model is that the tumours differentiate to neuroendocrine prostate carcinoma. So our group, we're interested in hereditary variation and how that influences prostate cancer. And previously we did a study, a TRAMP Cross F1 study, and we found that hereditary germline variations can modulate aggressive prostate cancer in these mice. And here we see that depending on the paternal strain, we can see a difference in primary tumour burden in the blue here and also lymph node metastases. And this led us to go on to develop a TRAMP F2 intercross study in a few of these strains where we did QTL mapping and we identified several novel prostate cancer susceptibility genes and also protector genes. But the limitations with these studies is that we had low genetic variation within these F2 intercrosses, and that gives us low mapping resolutions, and large modifier locus confidence intervals. So we wanted to use a mouse cross that would give us better mapping resolution. So this led us to the diversity outbred mouse. I'll just give you another look of the slide. I've seen this a few times as well today. So the diversity outbred mouse is generated from 8 founder inbred strains and is outbred to generate a highly genetically diverse outbred mouse population. And it has the potential to give us sub-megabase mapping resolution. So our experimental strategy was to use the male JDO mice, and we used a total of 192, and we crossed those with the female tramp mice. And we generated about 493 tramp cross DO F1 positive progeny. And then we used these mice and quantitated the phenotypic traits of aggressive prostate cancer, that is the primary tumour burden, shown here, and also the metastasis count in the lung, liver and lymph nodes. So once we had all the phenotypic data, we moved on to our cross-species integration. And the first part I'll talk to you about is the mouse study. We took the genomic DNA from the tail and used SNP genotyping, and we mapped the QTLs. We also extracted RNA from the prostate tumours and performed RNA-seq quantification and mapping of cis eQTLs to give us a list of mouse candidate genes. The second part of the study was taking these mouse candidate genes, using the human orthologs, and looking for association between variants of aggressive prostate clinical variables and also any association between gene expression levels and aggressive prostate clinical variables. And then this would give us filter down a list of high priority candidate genes that we could then go on to functionally validate in vitro and in vivo. So first what we found was on chromosome 8, we see a distant metastasis-free survival locus. And that is looking at mice that harbour a distant metastases versus mice that have no metastases. And we see here a peak linkage on chromosome 8 at about 47.8 megabases. So we then went on to try and identify the SNPs that were driving this linkage, and we found here 671 significantly associated SNPs that were associated with distant metastasis-free survival. And finally, in the mice, we looked at, we filtered down to the candidate genes through transcriptomic analysis. We performed RNA-seq on 195 prostate tumours from the TRAMP-DO mice. And we wanted first to identify cis eQTLs, that is transcripts within 1 megabase of our metastasis-associated SNPs. And we also looked at trait correlation analysis. And our final gene list was those that harboured both a cis eQTL and a distant metastasis-free survival correlation. And what we found was that there were 11 prostate cancer metastasis candidate genes listed here on the left, and all of these had both a cis eQTL, significant cis eQTL, and a significant association with distant metastasis-free survival. So next we looked at the human orthologs in the GWAS and prostate cancer gene expression datasets. And what we found was there was evidence for association with aggressive prostate cancer for 5 of the genes in human GWAS. We utilised 2 publicly available GWAS cohorts with over 1,200 cases and 2,500 cases. And these were the genes that we found to be significantly associated with clinical traits of aggressive prostate cancer. And next we used Seed Bioportal and found 3 prostate cancer data sets that had sufficient expression and clinical data. And we found association of 3 genes in human prostate cancer data sets, caspase-3, SEMPU, and RWDD4. We further went on to look at the effect of dysregulation of these 3 genes and their association with survival. What we see here is in this first data set, out of almost 500 patients, we see a total of 13 patients that had dysregulation of 1 or more of these 3 genes, and we see a significant effect on both disease-free survival and overall survival compared to patients who had normal expression levels. We also saw this in the second data set of 150 cases, where 38% of the cases were dysregulated in one or more of the genes, and there was a significant effect on disease-free survival compared to patients with normal expression levels. And finally, in the third data set, we saw a total of 7% of people had dysregulation of one or more of the genes out of 545 patients. And again, we saw a significant effect on metastasis-free survival and overall survival. So now that we'd sort of filtered down to our 3 candidate genes, we then wanted to go on and functionally validate these both in vitro and in vivo. We decided just to focus on SEMPU and RWDD4 since caspase-3 is a well-studied apoptotic protein. So we transfected SEMPU or RWDD4 into 2 cell lines, PC3 and LN caps, and then we use these in vitro assays and in vivo assays, which I'll talk to you about now. So in the in vitro functional validation, we first performed a soft agar assay, and this is an anchorage independence assay and is indicative of malignant transformation in vivo. And what we found was that both in PC3 and LN cap cell lines, overexpression of SEMPU significantly increased colony formation. And overexpression of RWDD4 was only found to increase colony formation in LN cap cells. Next, we performed a Transvaal invasion assay, and this highlights the cell's ability to invade a collagen membrane and is indicative of metastatic potential in vivo. And what we found was that cells overexpressing RWDD4 in both LN cap and PC3 cells were able to significantly increase cell invasion across the membrane compared to control, although we saw no difference with overexpression of SEMPU. So next we performed our in vivo assays. This assay is an intracardiac metastasis model, and what this measures is the potential for these cells to metastasize to distant sites. We injected PC3 cells that were co-expressing a luciferase gene as well as the gene of interest, injected them into the left cardiac ventricle, and then imaged them over 6 weeks to quantify the bioluminescence. And as you can see, what we found is that mice injected with cells overexpressing RWDD4 significantly increased metastasis compared to control, although overexpression of SEMPU didn't reach significance at 0.068. We see a trend happening here. So because of the inconclusive nature here, we decided to perform a flank assay with these with cells overexpressing SEMPU. So here we injected them subcutaneously into the flanks of nude mice and then measured the tumour size weekly. And as you can see, after only 3 weeks, these cells had a significant increase in both tumour volume and also tumour incidence. All of the mice here had tumours, whereas only a couple of the mice in the control group had tumours. And finally, what we wanted to look at was overexpression of RWDD4 and SEMPU and how they affect pathways related to tumorigenesis. So we took 4 to 5 clonal isolates of both RWDD4 and SEMPU overexpressed in PC3 cells and performed an ingenuity pathway analysis. And what we found were that several pathways related to tumorigenesis were not surprisingly affected. And but of keynote here is that MYC-N, which was found to be an upstream regulator of SEMPU, has recently been found as a key driver of neuroendocrine prostate cancer. We also found that overexpression of RWDD4 modulated the BRCA1 DNA damage response. Previously, a study has shown that there's a protein-protein interaction between BRCA1 and RWDD4. And finally, what we were able to show was that overexpression of RWDD4 upregulated multiple centromere components, including the SEMPU, which was our other candidate gene. So future work will go on to look at the interaction of these genes and how they affect development of neuroendocrine prostate cancer. So just in summary, the study has shown how integration of a system genetics approach using a cross-species analysis can identify novel hereditary variants that influence metastasis of prostate cancer, that is caspase-3, SEMPU, and RWDD4. And this study also adds insight into the molecular mechanisms of aggressive versus indolent disease and could ultimately lead to improved outcomes in high-risk prostate cancer patients. So I'd just like to thank all the Crawford Lab members and also all the collaborators involved in a lot of the work. Thank you.
Let's have one question.
I have one question. It's two parts. I guess I'll start with the first part if there's time to do the second part. So the first part, you did the EQTL mapping on tumor tissue, right?
Yes.
So I'm just wondering, it seems like there's two possible inferences there. One is it's a normal EQTL like we like to study, or the expression of the gene of interest is modified by the fact that you're in a tumor environment, and the QTL is really a QTL for creating a tumor, which then changes the tumor environment, which then changes the gene expression. Do you want to comment on that? Does it matter to you? Is one better for you or the other?
Not really sure. I haven't really thought about that.
So the other part I was wondering is, maybe you could clarify something for me. You did your QTL mapping, and a number of genes met the initial criteria in mouse, and then a subset of those genes also met the criteria in human.
Yes.
So it seemed like a simple scenario would be you think one gene matters in the mouse and that same gene matters in humans, but it's like something like 3 and 3 in both. I'm wondering how common of an outcome you think that is. I mean, it almost suggests that there's like a co-localization of genes in the genome of similar function, and that's why they're all affecting these pathways in both mouse and man.
Yeah,
is this a common outcome in the kind of studies you do?
I'm not too sure. I'm a first year and I'm sort of new to genetics, so I'm not really sure. But it's quite possible that that's the case. Yeah.
Okay.
Let's thank Jean, our Vern Chapman lecturer, Joe Nadeau, and all the speakers from this session.